We started our laboratory activity by placing a marijuana stalk in a glass slide and place it under a microscope. It has a rough, thick, dark green color and is covered in tiny hair-like structures. Next is the tuba-tuba leaf that has a vivid shade of light green color with a pale yellow leaf vein and leaf stem. Next is the papaya leaf that shows a dark green color, a pale yellow leaf vein, and a rough texture. Next is a dried marijuana leaf. It shows a thin, pointed leaves covered in tiny hair-like structures. The edges of its leaf resemble a cat's or bear's claw. This close-up of the paragis shows its rough texture. The ends have symmetrical straight lines and a rich green color. Marijuana seeds can be observed to have a dark brown color, with a crack in the middle, and they resemble a teardrop shape. Additionally, the seed exhibits a tiger-like pattern throughout the whole seed. The first thing we did in doing the paraffin test was to prepare the materials, such as the double-action handgun, the paraffin wax, and the bond paper containing a traced outline of a hand. The instructor ensured that the handgun was not loaded. After providing proper instructions, the instructor proceeded to load the firearm and hand it over to the subject.
I am ready.
After the subject fired the handgun in the firing chamber, he proceeded to wash his hands with running water without touching any surface. We then heat the paraffin wax in a can until it melts. With the use of tongs holding cotton, the melted paraffin wax was poured over the subject's hand until the entire hand was covered. We also placed cotton of the same size as the subject's hand, then applied a second layer of paraffin wax. We made sure that the wax was smooth and evenly distributed. After waiting several minutes for the paraffin to cool and harden, the subject carefully removed his hand from the cast. The instructor carefully removed the cast from the paper and applied diphenylamine reagent onto it. While the instructor poured the diphenylamine reagent, we focused on looking for blue specks as an indication that the subject had gunpowder on his hands. For our glass fracture activity, we used a broken jalousie and applied a coat of varnish onto it. We then placed the glass into a piece of cardboard and secured it with tape. We then placed the glass covered with varnish and tape inside the firing chamber. The instructor ensured the handgun was unloaded before loading it with a .38 mm bullet. The instructor then fired the handgun, aiming at the glass inside the firing chamber. We took out the cotton from the chamber to retrieve the glass that had been shot. Upon closer inspection, we observed lines radiating from the bullet hole along with circular cracks surrounding it. For this activity, we collected different kinds of animal hair, human hair from multiple body areas, and various fabrics. This sample shows a female's armpit hair, which has an uneven, rough texture and lacks a visible medulla. This is a female's pubic hair, which has an uneven surface and a thin, interrupted medulla. The hair from a male exhibits an uneven texture with a light brown medulla. The beard of a male displays a coarse, uneven surface with a thick medulla. Under a microscope, a male's armpit hair has a structure similar to his beard, but it only differs in length. This is a male's mustache, which has an uneven, thick, and coarse structure along with a thin medulla. A male's pubic hair has a thick structure but lacks a medulla. This is a pig's hair, which has a coarse, thick structure and a blurred, wide, white medulla when viewed under the microscope. This is a cow's hair, light in color with a thick structure and a brown fragmented medulla. The dog's hair has a smooth texture and a dark colored edge along with a wide, continuous, and well-defined dark medulla. This is a cat's hair. It shows a very dark color, uneven cuticle, and a thick dark medulla. This is a silk fabric under a microscope. The fibers are wavy, flexible, and very thin. This is a wool fabric. It shows a bundle of intertwined fibers. The individual fibers appear relatively fine and curly or wavy. Next, we proceeded to burn the animal and human hairs, starting with the goat's hair. When it was burnt, it produced a strong odor. Next, we burned the dog's hair. It produced a light burnt odor, milder than the goat's hair. It burned quickly and left a fine ash. We also burned the cow's hair. It gave a milder burn smell compared to goat and dog's hair. The hair was thick and coarse and became dark brown and brittle. In this activity, we used a blood lancet to prick ourselves, collected the blood droplets, and placed them on a glass slide. Next, the instructor added a drop of anti-Sera A to the blood droplet and mixed it. He also added anti-Sera B to the blood labeled B and anti-D reagent to the blood droplet labeled D. He repeated the same process with other blood samples so we could determine our blood types. After a few minutes of adding reagents, agglutination could be seen in the blood samples, displaying different outcomes. This blood sample is identified as type B positive. This is also a type B positive blood, and this is an AB positive type of blood. The blood on the fiber cloth was a pig's blood added with benzidine reagent. We also examined human blood mixed with saline solution under a microscope, and this was the result. This is human blood with Tekman reagent, human blood with Takayama reagent, pig's blood with Tekman reagent, and pig's blood with Takayama reagent. We first collected fresh semen, placed it on a glass slide, and examined it under a microscope. Millions of sperm cells are seen alive and active. A dried semen stain on a fabric was collected, mixed with saline solution, and observed under the microscope. It shows that only a few sperm cells remained intact and alive. Picric acid was also added to the large semen, which was then observed under the microscope, and this was the result. It shows the presence of numerous small structures that appears to be the crystals. In this activity, we began by preparing all the necessary materials. Next, we filled the shoebox with sand and soil to create a surface where we could step and leave our footprints and shoe prints. Then, we sprayed the impressions we made in the sand and soil with plastic varnish to help preserve and harden them. We also sprayed oil to the foot and shoe marks imprinted in the sand and soil. Next, we put the plaster of Paris in a Ziploc bag, mixed in 500 ml of water, and squeezed the bag until the mixture thickened into a paste. We then poured the mixture over the impressions and smoothed it with a stainless steel trowel. We added an aluminum screen for support, then poured another layer and spread it evenly. Next, we placed the index card containing the evidence details. After waiting for about 10 to 20 minutes for the cast to harden, we carefully took it out of the box to check the results. We also washed the casting under running water to remove any sand or soil so the details of the foot and shoe impressions could be seen more clearly. For this activity, we prepared the sandpaper number 100 for rough sanding and sandpaper number 1000 for smoothing and finishing. We also have a wrench tool that we will use to remove specific details. We began the process by applying sandpaper number 100 to the wrench and sanding the letter engravings thoroughly for several minutes. After that, we switched to number 1000 sandpaper to make the surface smoother, sanding for a couple of minutes until the letters looked a bit faded. Next, the instructor applied drops of muriatic acid to the sanded engravings and wiped it with a dry cloth. After putting drops of muriatic acid on the sanded area, the engravings on the wrench became lighter. After that, the instructor put a drop of picric acid on the same spot where the muriatic acid was placed, and wiped it again with a dry cloth. Finally, once we wiped the picric acid from the engraved section, the markings on the wrench reappeared and looked like their original form. The reaction occurred in only a split second. We began the activity by gathering all the necessary materials. This included various brands of bond paper, different types and brands of ballpoint pens, as well as several markers and highlighters. We also prepared 2 beakers and filled them with tap water. Salt was added to one of the beakers and allowed to dissolve completely. We added a few drops of ink into the beaker with tap water. We also added a few drops of ink into the beaker with salt water. At first, the ink didn't spread. It stayed thick, clumpy, and remained on the surface. After some time, it slowly dispersed and formed irregular broken patches. The next thing we did was test each highlighter brand and color by making a large dot on paper. We prepared 2 separate sheets and applied the highlighter samples on both. Next, we dipped the 2 papers with highlighter ink samples into separate beakers. The beaker on the left contained ethyl alcohol, while the beaker on the right contained tap water. After a few seconds, the paper soaked in ethyl alcohol showed obvious changes. The alcohol was absorbed quickly, making the highlighter ink spread out and causing its color to change fast. The pink staphylococcus ink beside the yellow ink even started to fade, while the other brand's colors stayed mostly the same. In contrast, the paper dipped in tap water absorbed the liquid more slowly, and the ink mainly spread downward along the surface of the paper. After completing the ink analysis using different highlighter brands, we proceeded to conduct the same test with various markers. We repeated the same steps used for the highlighters. We placed a dot on the paper using each marker, then did the same on another sheet, alternating the marker inks used on the first paper. We then dipped the papers into the beakers containing ethyl alcohol and tap water, and waited patiently while carefully observing the results. After a few minutes of observation, it became clear that the marker ink spread widely and even moved upward on the paper. Meanwhile, the paper dipped in tap water kept the ink mostly intact, showing little to no visible change. We also tested various brands and colors of ballpens. We placed a dot on the paper using each ballpen, then repeated the process on another sheet, alternating the ballpens used on the first paper. The next step we did was dip the sheets of paper with ballpen dots into two separate beakers, one containing ethyl alcohol and the other filled with tap water. After that, we waited patiently and closely observed the changes that occurred. After waiting for a minute or two, the inks in the tap water beaker showed changes more quickly compared to those dipped in ethyl alcohol. After a few more minutes, both beakers showed clear results. The paper soaked in ethyl alcohol also caused the ink to spread upward, but not as much as the paper in water. The water made the ink spread even farther and caused the colors to fade a little. After analyzing the inks from different brands and colors of highlighters, markers, and ballpens, we then moved on to testing various brands of band paper. The first thing we did was place our fingerprints on all the different brands of bond paper we prepared. We then held each bond paper with fingerprints over the metal can using metal tongs, allowing the violet smoke from the iodine fuming to rise. This reaction helped make the fingerprints appear on the paper. We did the process on all brands of bond paper we have. After the iodine fuming test, we compared the results from all the brands of band paper that were exposed to the iodine smoke. In this activity, we prepared bottles of different types and shapes. First, we used a rum bottle, filled it with ethyl alcohol, and shook it thoroughly to spread the alcohol up to the rim. Next, we ignited a match, placed the flame at the rim of the bottle, and observed how quickly the flame traveled to the bottom. A juice bottle was used as well, following the same steps, adding alcohol, shaking, and igniting it. The last material we used was a 1.5-liter soda bottle, repeating the same steps of adding ethyl alcohol, shaking, igniting, and observing the results. Here, we can observe that the flame quickly traveled to the bottom and the flame at the rim resembled a torch.